This 5-minute audio slide presentation will provide an overview of the paper Mechanisms Underlying the Formation of the Amygdalar Fear Memory Trace, a Computational Perspective. Experimental studies in the Joslin Lab in Toronto have shown that 25% of rodent lateral amygdala cells are potentiated after Pavlovian fear conditioning. This 25% set of potentiated cells is termed the fear memory trace, since deleting them eliminates fear. They have also found that excitable cells have a better chance to be in the fear memory trace. More interestingly, they found that if the number of excitable cells is increased prior to fear training using KREB, the size of the fear memory trace remains unchanged. They hypothesized that competition among the pyramidal cells might be the cause. However, the underlying mechanisms involved in this competition are unclear. We used a computational model of the lateral amygdala to determine the underlying mechanisms involved in the competition. The model has 1,000 neurons and has the structure shown in Figure 1. We had 5 model cases with different percentages of excitable cells in the lateral amygdala, starting with the control case, which had 50% highly excitable cells. These cases are labeled as KREB cases in the figures. Each of these model cases was subjected to Pavlovian conditioning using tone and shock as shown in the figure. We checked, does the fear memory trace remain constant in all 5 cases? If so, what underlying mechanisms made them remain constant? We found the following. The principal cells that exhibited significant increases in firing are labeled plastic, and they were of 2 types, LP and TP cells. Those that did not potentiate are labeled as non-plastic. First, the tone responses of LP, TP, and non-plastic cells remained within biological ranges in all cases, as shown in Panel A of Figure 2. More importantly, the number of LP cells remained approximately constant, so the first prediction is that LP cells store the fear memory trace. Moving on to the underlying mechanisms involved in the competition, the model provided the following predictions. First of all, among the type B and C PNs, only those that received both tone and shock or only tone inputs participated in the competition. Within this competition group, LPs received significantly more excitatory inputs from other PNs than TP and NP cells. Most importantly, as competition increased, the network selected PNs to be memory cells that received the fewest disynaptic inhibitory connections from other PNs in the competition group. Those trends were shown in Figure 3A and B. A separate analysis of winners and losers showed the same trend. Figure 3D and E showed our insights into the formation of TPs. Compared to LP cells, TP cells receive significantly stronger monosynaptic inhibitory connections, both in numbers and an increase of synaptic strength. As a final model prediction, Figure 3F shows that the level of inhibition in the system controls the size of the fear memory trace. We found that only a particular group of highly excitable principal neurons compete during fear memory formation. Of these, synaptic competition selects a subset using inhibition as the dominant mechanism. The insights related to the underlying mechanisms of memory formation have potential applicability to memory formation in other cerebral networks. Finally, the finding that the level of intrinsic inhibition has such a profound influence on the integration of projection cells into fear memory traces has potential clinical relevance. It implies that slight reductions in the amount of intrinsic inhibition could allow the number of cells encoding fear memories to increase, compromising their stimulus specificity.